The new finding of severe dieback caused by Lasiodiplogiteobrome is becoming a major constraint to cacao production here in the Philippines. Early detection of this pathogen will allow management of disease infections and minimize the risk of the spread of disease. So traditionally, plant pathogen detection requires isolation and morphological evaluation that may take several days. In this study, two detection methods were developed, and Phytophthora palmivora was also used as non-target pathogen to determine the specificity of the developed method. So first is the use of functionalized magnetic nanoparticles or MNPF1 for rapid detection by sequential magnetic cell capture. So blank and spore suspensions of target and non-target were mixed with MNPF1 and incubated for 10 minutes before placing in a magnetic rack. After the magnetic separation, these images were taken and analyzed using ImageJ software to determine the matting area in the presence and absence of spores. The matting area of blank and samples were analyzed using one-way ANOVA and LSD test at alpha 0.05 or at 95% confidence level. And a significant mean differences were observed between blank and samples. The MNP zoospore complex and blank were also resuspended in phosphate buffer and spread plated onto HSPDA. The confirmation of capture was then determined after 3 days of incubation. So from these results, it was proven that MNPF1 can indeed capture fungal cells and can be used for rapid diagnosis. However, using this method alone, identification of specific pathogen is not possible since both target and non-target were captured using MNPF1. Thus, a sequence-specific detection method was developed using citrate cup gold nanoparticles and unmodified oligonucleotide probe. The hybridization of the probe and target DNA resulted in formation of double-stranded DNA hybrids. The adsorption of the hybrids to gold nanoparticles prevented aggregation upon addition of salt, as can be observed by no change in color. In contrast, gold nanoparticles in the absence of hybrid DNA undergo an abrupt observable color change caused by aggregation upon addition of salt. So from these results, the method showed a capability to differentiate the presence of Lasiodiplogia theobrome from another fungal pathogen visually. However, it should be noted that optimization and validation of these detection methods are still recommended for its potential to be fully realized.